Thank you for being here. This is welcome you for the Center for Causal Discovery. As I think most of you know, the Center is sponsoring this lecture series in order to recognize distinguished leaders in the field of causal modeling and discovery, and to provide a forum here for us to let us know about some of the cutting-edge research and developments that are going on in the field. So it's my pleasure in particular to introduce Professor Joris Moij as our distinguished lecturer today. Dr. Moij is an associate professor at the Informatics Institute at the University of Amsterdam, which he joined in 2013. And in 2007, he received his PhD in mathematics and physics with honors from Radboud University in Nijmegen in the Netherlands. He then pursued causal discovery research as a postdoc at the Max Planck Institute in Biological Cybernetics at Tübingen in Germany, and then as a postdoc at Radboud University. His research interests include causal discovery, both fundamental and questions and applications in biology, approximate inference, and machine learning methods more generally. In recent years, he's been quite active in research, publishing, and other contributions in, numerous cutting-edge topics in causal modeling and discovery. In 2016, his team won the CRM Causal Inference Challenge, and he's won a number of additional awards for his work. As you can see from his title here, his talk today will be on validating causal discovery methods, a very interesting and important topic. Dr. Moij, we're really delighted to have you here. Welcome. We're looking forward to your talk, and I'd like to welcome you.
So thank you very much for the kind introduction and for inviting me here. I'm really excited to be here. This must be now the world's leading center on causal discovery, I guess. I will indeed speak on causal discovery and more in particular on how can we validate causal discovery methods. Right. So as you all know, establishing causal relations from data, causal discovery is one of the fundamental tasks in science. And since the pioneering work by Pierce and Fisher, the gold standard for causal discovery is a randomized experiment. And more recently, causal discovery methods from purely observational data have been developed. For example, well, it all started out with the work by Spertus, Weimer, Shines, Pearl, and others, some of whom are in this room. That was in the 90s or so, 30 years ago. And these ideas have inspired causal discovery methods that combine observational and interventional data in various ways. And the question I would like to focus on today is how well do these alternative causal discovery methods work in practice? Can they, do they provide any useful alternative to the gold standard? I don't know exactly how much you all know on causal discovery. I have a few introductory slides. Let me just go through them quickly in order that so that we are all on one page. So first of all, causal discovery by experimentation. I guess you're all familiar with this idea of a randomized controlled trial, so-called A/B testing in engineering. Gold standard. Here, for example, in a clinical medical setting, we have a group of patients. We want to find out whether a drug is a cause for an outcome, for example, recovery from some disease. So we split the patients into two groups randomly by, say, a coin flip. And one group is the control group. They get a placebo. The other group gets the actual drug. And if there is a difference in distribution for the outcome between both groups, then we can conclude from this directly that the drug causes the outcome, say, recovery. A very simple but powerful idea. And the whole, let's say, the evidence-based medicine nowadays is based on this approach. I always took this for granted myself because it seems like a simple idea. But you can also think about it in terms of graphical causal modeling. And you can actually prove that this conclusion, this idea makes sense in the following way. Suppose that the treatment and the outcome, C and X, can be modeled with a structural causal model. And then if we make these assumptions that the outcome does not cause the treatment, which is very plausible because the outcome happens later in time than the treatment. So as long as we don't believe in time travel, this is well motivated. The second assumption is that the the outcome and the treatment are unconfounded. So this is where the randomization comes in. If we assign treatment by a coin flip, then it would be very unlikely that there's some latent common cause that determines the outcome of the coin and the outcome for the patient. So the only thing I could think of for such a confounder could be the hand of God, for example, that interferes with our experiments. But, if we dismiss that, then this is also a good assumption, assuming we do proper randomization. And finally, we have to assume that there's no selection bias, so we should measure and analyze all the samples, basically. And under those four assumptions, if we see a dependence between treatment and outcome, then the treatment must cause the outcome. And the proof is really simple. You can just draw the 8 possible causal graphs on these 2 variables. You can include feedback cycles if you want to. They're ruled out by these assumptions again. But if you sort of the only 2 models that are actually possible under these assumptions are these 2, and that's the simple proof. Causal discovery from observational data, and there are different ways of doing this. It all started with constraint-based causal discovery methods. Here's a very simple example of this idea. Here we have 3 variables, x, y, and z is color-coded. It's binary, blue or red. And if you look at this data, you see certain dependencies and independencies. For example, x is independent of y, but x and y become dependent when we condition on z. And you see this whole pattern here. The question is, what is the causal relation between x, y, and z? Well, that's an easy question, I guess, for this audience. Well, okay. A hint is that you can assume several things like causal Markov assumption, causal faithfulness, the absence of confounders, the absence of cycles, the absence of selection bias, and the absence of measurement error. And under those assumptions, we can actually conclude that x must cause z and y must cause z, and x and y are causally unrelated. So this is very intriguing. We don't have to do an experiment. We get our causal conclusions just by observing under these strong assumptions. Now, over the years, many methods have been proposed for causal discovery from purely observational data. There are too many to mention. Wrote down a few. Exploit conditional independence patterns, non-independence constraints like Fermat constraints. You can try to exploit those. There are likelihood-based approaches, and there are other approaches that are not looking at independencies, but try to exploit restrictions on the functional causal relations or on the noise distributions. And then we can combine these ideas. So maybe we can actually combine data from different contexts. So for example, different interventional data sets. And also there, many methods have been proposed. This is a table I recently made. Also, this is not a complete list, but as you can see, there are many methods that have been proposed with different features. So basically, we have to our disposal a whole range of causal discovery methods. And they have all been shown to work on simulated data if their assumptions are satisfied. But do they actually work in practice? Question. If you try to provide convincing validation that these methods or one of these methods works in practice, this is actually challenging. What you need is a reliable external ground truth that comes from an external data set or from domain knowledge, which is typically a summary of many independent experiments that have been done. Or an alternative is if you use a held out test set. For that, you need enough interventions. And in addition, you need enough prediction tasks to outperform simple baselines significantly, and even after you correct for multiple testing, as we will see. So, and this I would like to pose this claim here as sort of a discussion item. I hope that you can prove me wrong. I will back up part of this claim in this talk later today. I would claim that alternatives to the randomized controlled trial idea have not had big impact yet in other scientific disciplines because of a lack of convincing validation so far. Maybe we can discuss it later at the end of my talk a little bit more. I will focus my talk on this part. First part of the claim is just my personal impression that I have. So lack of convincing validation so far. So question, success stories. What can anybody here tell me a success story of causal discovery from purely observational data? I've come up with some success stories myself, but with this audience, I thought it's really nice to get some more input on this. So if you know anything, then please.
Well, what do you define as a success story? Not a success.
A success story, like.
I mean, success. I mean, does that?
So.
I mean, I'm serious because
so, yeah.
There's different degrees of what that means.
Of course. Let's just start easy and say anything where you think, or where people think that people have shown that this works on real data and is perceived as a success in the community. Early detection of cancer. Is there any specific paper or that you're referring to? You are working on it. Okay, so it's a future success story. Or so this will become a success story. Great. but that's maybe in the future. Sorry? Is that IDA applied on? Sorry?
It was C-star.
C-star.
Bootstrapped IDA.
Bootstrapped IDA. Okay. I'm not familiar with that particular application. Or is that in the Annals of Statistics paper about IDA?
No,
It was later. No, that was later. Yeah. Maybe I should add it to my list.
They did an experimental.
Yeah. Okay. Yes, almost, although it's not from purely observational data. The next question will be more broad, and then that would be a good one. Okay, according to the main experts. So can they convince us that it's really convincing? Uh-huh. Okay, that sounds like a success story indeed. I don't know that, but so if you I can't remember all this, but maybe for my next talk, if okay, so what paper is that, or what? So what paper, which year is there? Okay. So maybe if you could, I can't remember or write down this all. So if you send me an email afterwards, that would be great.
Will you maintain the list?
Yeah, why not? Why not? Did you want to say something? Or bring something? IDA, yeah, the yeast data. Yes, I will come to that. Yes. Yeah. So the three things that came to my mind were trigger algorithm on yeast genomics data. So that's LCD related algorithm. Ken and the others, Genome Biology 2007. IDA on the yeast data, Nature Method paper 2010. And the Tübingen cause effect pairs, it's by some perceived as a success story. Please feedback I get on it. I'm not so sure myself actually, but I put some question marks here because we'll take a closer look whether it's really successful or not. Going back to your question, what does it mean, success? Okay, more broadly, maybe there are more convincing success stories even when we don't look at from purely observational data, but more broadly, if we are allowed to combine it with interventional data. Any other success stories? We already heard the flow cytometry data from Sachs and others. I will add
Well, so
2 more here.
we're stuff with the hippocampus.
Some degree of covering what you're at.
From fMRI data.
From fMRI data.
Recapitulating known relationships in the hippocampus.
Yeah, I've worked on something very small in the past, and then to me, I was struck that the domain experts were always saying, this is an interesting model. Yeah, that looks plausible. Even when the next week I came.
These were relationships from known other scientific.
Okay, great. Yeah, so yes, that maybe actually I would be fun to maintain a list like that. So if you send any inputs, make a list. So I also have ICP, invariant causal prediction, on yeast gene expression data. And more recently, I'm not sure if you're familiar with this work, but this is LCD and backshift evaluated mass cytometry data. Oh, she was here. Great. Yeah. Great. Okay, so a few success stories are around. Let's look a little bit closer at some of these success stories. So I will. Okay, I'm already going much slower than I anticipated. Nice interaction. I have basically 3. So maybe we can make this interactive. 3 parts. One is on yeast expression data on IDA and yeast. Another part on the cause-effect pairs that I could talk about a little bit. And then on reconstructing signaling networks from flow and mass cytometry data. So if you have any preferences, then let me know, because then I'll reserve some more time for it. I have a preference myself for the yeast gene expression data, and then we'll see how much time is left. So IDA, Nature Methods paper. Back then, in 2010, they had this nice ROC curve when they evaluated the PC algorithm combined with a clever way of using parent adjustments to get bounds on causal effects. And they evaluated this using this interventional yeast gene expression data. And they showed that they could predict the strongest causal effects much better than random guessing or regression-like baselines. So I was very happy back then. I thought, great, convincing proof that constraint-based causal discovery really works. And I also thought maybe even a simpler method would also work, like maybe something like LCD or looking for Y structures, or something really simple would also work. So that's what was when I tried to sort of reconstruct what they have been doing, and I started looking into this data. That was already maybe 4 or 5 years ago, and it has been a somewhat painful process for reasons that I will explain. So first, let me explain the data. We have a micro-array data for 6,000 or so yeast genes. Oh, by the way, they did it on this Huxedel data sets from 2000. Meanwhile, we have a newer data set from Kemmeren and others, 2014. This is much better quality, much more modern, and it's also larger data set. I'll focus mostly on this one here. We have observational data where they measured gene expression levels, population gene expression levels for yeast, and they repeated it about 250 times, which is a lot for biologists. They usually do it once or twice. Here they did it 250 times. And then there's interventional data where they knocked out a single gene, one at a time. And then after each knockout, they also measured the response of the gene expression levels. And so we have one interventional sample per gene knockout. But we have 1,500 or so of them. And the task is here, can we predict from the data which gene expression level change when a certain gene is knocked out? Simple causal prediction task. So here are a few samples from the data sets. Here are scatter plots of 2 genes. And here you see a Fuchs version and here the Kemmeren version. These two look pretty similar, but they can also look different, like here in this example. Here you see with the blue dots, you see the, I should try and use this pointer. The blue dots are the observational data points. The red ones are the interventional ones. And this green cross here That's what you get if you knock out the gene that's on the x-axis. So if you knock out this gene YMR275C, then actually the gene expression level of that gene is reduced significantly. So yes, the knockout was successful. It was turned off. It wasn't expressed anymore, this gene. And basically, to interpret these values, you should know that we don't measure the absolute gene expression levels, but it's always the difference of the log ratios of the gene expression levels from a wild type with a wild type, so from genetically identical populations of yeast cells. So in this particular case, we also see that knocking out this gene, YMR275C, doesn't have a strong effect on the gene expression YPL142C, because, well, it's slightly different from 0, but it's within the natural fluctuations in the observational distribution. So here, I think we would all agree that this gene doesn't have an effect on that one. Maybe it's different here in this case. So we can use this data to define our own ground truth for validation. They did in that paper. Now, and we have to somehow, previous example, we did it by eye, but we want to do this for all million gene pairs, so we have to automate it, so we have to find a good score for the causal effect strength. And there are some natural scores that you could look at. You could just look at the absolute effect when you knock out a gene, so just the difference between 0 and gene expression level after the knockout. But you can also normalize this with respect to the observational standard deviation. Alternatively, you could look at the slope of this line, which would be sort of corrected for the self-effect. How much is the gene expression level of the gene that is knocked out actually changing? This would be more like what is in the literature known as the causal effect. It's the slope of this line. And you can also express that, in different units by normalizing it with respect to the observational standard deviations, right? In their paper, they use something which is slightly different, but quite similar to this measure here. There's something called strong intervention effects that I won't explain now. You can think about it as a kind of discretized version of this absolute normalized measure. Okay. Now, typically, we are going to draw ROC curves and calculate AUCs to measure the performance. So what we will do is we'll just use the top, say, 5% of the strongest effects, according to one of these measures, as the causes, and the others are the non-causes. So, by the way, we only do this discretization because this allows you to draw an ROC curve. If anybody knows a nice alternative where we don't have to do this, an ROC curve, then we would be interested in that. So, but anyway, this is also what they did in the paper. We choose this theta somewhat arbitrarily to define a binary ground truth. Actually, if you plot these ground truths now using different measures, versus if you compare these ground truths, so for example, this is the absolute one, this is the relative normalized one. You look at the top 5% of each, then rather than a nice diagonal that you would maybe hope to see, you see this, which already indicating that the choice of this ground truth measure may have quite an impact on the conclusions. So this is first quite important empirical observation to make. Now we're going to compare with some baselines, Pearson correlation, Spearman correlation, some regression methods. Here I always indicate the units that we used. And also some very simple baselines, which are basically random guessing, but then scaled with the standard deviation, observational standard deviation, sigma j of the effect, or sigma j divided by sigma I. This may seem ad hoc, but well, at some point we found these baselines. So they are definitely very simple non-causal baselines that we should be able to outperform. Right? And there this question of the units. So here, this has a unit, this prediction score has units of being j. So I indicated via this, and this one has j over I. And we do basically a five-fold cross-validation. We can use part of the data for predictions and another complementary part for testing and then cycling through. And if we do this, for example, for a ridge regression as one of our baselines, we get the following AUC results. So this is on the Kemmeren data where we take the top 10% as the true facts. And here you see, vertically, you see different ground truth measures that you could use. Horizontally, you see different ways of scaling the outputs, the predictions of ridge regression to get different units just by scaling them with sigma i's and sigma j's. And the first thing that you see is that the scaling has a large effect on the AUC. So here, for example, ridge regression, unscaled ridge regression, which gives a regression coefficient, which is in units j over I, has an AUC of 0.61 if we use this relative ground truth, which is also looking at the slope, which predicts this, uses the slope of this line, the ground truth measure. So this is interesting that the scaling has so much of an effect. So we argued that maybe we should, in order to avoid comparing apples and oranges, we should only use matching units. So if our prediction, if we use certain units for defining the ground truth, then we are saying basically our prediction task is to predict the strongest effects measured in such and such way. And maybe, sorry, the validation task is, yeah, then we should use that same measure for our prediction. So that's indicated with these black boxes here. So that already makes it, well, rather than looking at 25 of these numbers, we only have 5 to look at if we do that. And here you see what you see also the results for other baselines, including these random scaled baselines and also IDA on the left-hand side. Here it's unscaled, so here we might be comparing apples with oranges. Here on the right-hand side, we have scaled our predictions to get the correct units. And you see a more consistent pattern emerging here. Those first conclusion, scales matter a lot. They are dominant for your conclusion. This evaluation. And also we see that, well, we thought ridge regression was doing pretty well with an AUC of 0.61 using this ground truth score here. But actually, this random baseline, which was just sigma j multiplied with a random number to break ties, basically, is doing even better. And we were really surprised how this could be. We thought we would be measuring how well a method can predict causal effects. Yet a simple baseline that only looks at sigma j, not even at sigma, so the sigma. So it says basically that if gene J has a high standard deviation, then it's very likely that any other gene will cause it. That's basically the prediction method, and it does very well here. The explanation is actually, once you think about it, relatively simple. The causal relations are sparse, so for most of the gene knockouts that you do, there won't be an effect on xj. Basically, when you try to predict this value, the value of xj under knockout of I, then it's similarly distributed for most I as the observational value xj. Therefore, a good predictor for this absolute value is just sigma j. So maybe actually, after all, these ground truth scores, the unnormalized ones, are not so interesting because they are not really what we are after here. And okay, well, if you then don't look at these two rows here, then what's left? So let's zoom in a little bit. Here we see similar picture as in the IDA paper. looking at the strongest effects. And here we have different, well, so we have IDA evaluated with these different ground truth scores. And here you see, for example, first sight pretty impressive ROC curve. That's for this absolute ground truth score. So if we plot the baseline next to it, then it's no longer so impressive, right? So maybe we shouldn't look at the yellow and the red curves here. What's still remaining, however, are these nice ROC curves for the relative, what is it? The relative, if you look closely, sorry, relative normalized ground truth and the IDA ground truth that they used in their paper. That's still remaining here. And we don't have baselines that outperform those. At least not in the right units. But if you then look where does this nice causal signal come from, then it turns out that it's only due to 2 interventions on 2 genes. And actually, if you look closely, these genes, the knockouts of these genes were unsuccessful. So this is very suspicious, right? The knockout was unsuccessful, and yet they have many causal effects, apparently, according to these measures. So if we just remove these 2 out of the 234 interventions that are in the data, then, whoops, the whole signal is gone. Is it? Oh yeah, it's even doing worse than random guessing now. So I would say this is probably an artifact due to these 2 ineffective knockouts. Somewhere. Probably the artifact is in the reason that here, for delta xi is very small for these ineffective knockouts because the self effect is small, so we are blowing up anything by this small denominator. Yes, questions? The total effects, yes, total effects, yes. So it seems to be really an artifact here. And if we move on to another data set, Kemmeren, then we don't even have to remove ineffective interventions. Actually, here, I think all of the interventions are more or less successful in this data. And you see that none of the IDA ROC curves, or all of them, are outperformed by one of our simple baselines, either the skilled random guessing or, in this case, ridge regression. So I guess that's bad news for IDA on yeast data. There we are basically predicting sigma j, which is boring. But ICP seems to be doing significantly better than random guessing for this absolute normalized ground truth measure, and also for the strong intervention effects, the green and the blue one. And if we look more globally at partial AUCs of ICP, which is this column here, in comparison with the baselines, then we see in particular that for this absolute normalized ground truth measure, ICP is doing better than all our baselines. So that's promising. Another thing that's promising is that, remember that we had these arbitrary thresholds to binarize the ground truth. Now, hopefully, the fact that it's doing better than random guessing doesn't depend on this arbitrary threshold that we chose. And here you see the nice picture. You see nicely that if we take a stricter and stricter threshold, so only focusing in the ground truth on the strongest effects, then actually the partial AUC of ICP improves even. So it really seems to be focusing on and finding the strongest effects according to this evaluation. So I would say this is a success story. Well, you can find other baselines that still outperform it, but so far this is, I would say, a success story. The only maybe disadvantage of ICP here is that it's so conservative that it only gives you, say, 100 or 200 predictions out of a possible number of 1 million or so. So it's really very conservative, but at least it seems to be getting the few predictions it makes, seems to be getting it right. So can we do better than this? Actually, ICP just pools the interventional data, but we have more knowledge. We know for each intervention which gene is knocked out. So surely if we could use that information, we might be able to do much better. So I was hoping to use some constraint-based causal discovery methods to do this. And then I did this simple analysis of the data where basically for all gene pairs you can look, are they independent or dependent, observationally speaking? And are they causally related or are they not causally related by looking at the intervention data? Then you get these numbers by doing arbitrary thresholds here again. So the numbers are very much threshold dependent, but these fractions are less dependent on which thresholds you choose. One interesting thing is that the majority of the pairs that are causally related are actually independent in the observational data. So like 0.66 of them are faithfulness violations. I don't know where they come from exactly. It may have to do from the fact that actually one hypothesis is that we're looking at genetically identical yeast cells. So there's not much going on in terms of gene expression. So the only thing we are seeing is maybe measurement noise here. Could be an explanation. Anyhow, these are really many faithfulness violations to deal with constraint-based causal discovery methods. The opposite is also worrying. Even though we have measured almost all genes here, the majority of the pairs that are dependent, observationally speaking, they are not causally related. So either they are confounded or maybe there's some selection bias going on. And in principle, of course, we can deal with confounders, but maybe, well, if 90% of the pairs are confounded, then it starts to get a bit tricky, I guess. Yeah, so this is population data of yeast cells. Mm-hmm. So what we do, or what they do in order to get a measurement, they start with the yeast population. Maybe they do a knockout, or they don't do a knockout. Then they start with a small population. They have to let it grow for several generations until they have enough such that they can measure with microarray data. So if some gene knockouts, so in this process of multiplying and growing and surviving, at the end we see only the fittest yeast cells, and that might actually, suppose there's some redundancy in the how it works, and you knock out one essential part, or not completely essential part, but you knock out one part of the yeast that's important, then maybe it transitions to another way of operating, another metabolism state or whatever. And because you grow it for several generations, this may introduce the selection bias. Actually, we can only do the gene knockouts for which the yeast is survives at all. So just we have already selected on that, for example. And there might be actually very strong selection bias effects here. How to deal with them or handle them or even detect them, or maybe one cause why this could be a tricky data set. Cycles, that's also something I worried about, but if you look at how many the pairs are actually cyclic, then this is just a very small fraction. So maybe the assumption that there's a semi-Markovian model behind it might be reasonable in that light. So I don't know if there is much hope for constraint-based causal discovery in the light of those numbers. So some conclusions on this data set. Time is really running fast. So we've seen that how the data is scaled, something that we didn't spend much attention on originally, but it seems it has a dominant effect on our conclusions. So don't forget to take the scaling into account properly. For example, these unnormalized ones are not very useful to look at in this setting. One conclusion is that IDA does not outperform the non-causal baselines in our evaluation. So the conclusion by Maathuis, others, that IDA can predict the strongest causal effects by using only observational data, not tenable under closer scrutiny. ICP, on the other hand, outperforms all non-causal baselines for these two ground truth measures when looking at the few strongest effects, but not for the other ones. So if you're optimistic, you could say, this is a success story. If you're skeptical, you could say, well, why only this measure and not, say, the relative normalized one? If you're optimistic, you could call it at least a success story. Okay, now that time is running out, I have to switch either talking about cause-effect pairs or signaling networks from flow cytometry, mass cytometry. Who would want me to emphasize the first cause-effect pairs? Please raise your hands if you. Who would like me to emphasize the second part on signaling networks? Okay, that's quite clear. So, just very briefly, well, here there's nothing novel anyway that's not written in the paper. So, basically, we had this. So, very briefly, we had this benchmark data set collected over the years. We evaluated several methods, variants of additive noise models, and IGCI, information geometric causal inference methods, for those of you who are familiar with it. The results were kind of promising, it seemed to work, but there's the big but, it was not statistically significant, because in the end we ended up testing so many variants of these methods that if you just do a Bonferroni correction for all those, then your significance is completely gone. We would only have tested the best one. Actually, that was the first one from 2007 that we proposed, and had stopped by then maybe it would have been significant, but now. So short story, promising but not significant. So it's not a convincing success story, I would say. Yes, cytometry data. So the work by Kemmeren, Sachs and others, 2005 science breakthrough paper. This is flow cytometry data. There are different experimental conditions. Here, vertical, illustrated. They measured 11 phosphorylized proteins in single cells of human immune systems. They added different reagents in the different experimental conditions and with some known effects. And the task is combining this data and this background knowledge can we reconstruct the signaling network. I'll just give the results, skipping how you can do this. This is what they call in their paper the consensus network. This one here. Their own reconstruction is this one, where the deviations shown from the consensus are in blue and in red. So it's a very close reconstruction. And 2013, using a different method, also a likelihood-based method, I arrived at this reconstruction, which is also kind of close, but less similar to the consensus network. So I was wondering which of these three is now the most accurate description of reality. Well, okay, first of all, it's nice in the sense that there seems to be some causal pattern in this data that we can more or less reconstruct consistently. But if you zoom in and really want to say, is this now, which of these three is the best one, then becomes a little bit puzzling. So here you see these data, or part of the data. Here we have a logarithm of Raf versus logarithm of Mek, two phosphorylized proteins. Here I only plot two conditions, in blue the baseline, and in red the reagents called U0126 is added. So now, to the causal inference experts in the audience, what is the causal relation between Raf and Mek? Can you derive it? Can you see it by looking at this data from the information I gave you? Yeah, okay. So so you could try fitting an additive noise model or whatever, maybe to answer this question without knowing that. But indeed, if you have that background knowledge, it becomes easier. So a hint, UL126 inhibits Mek. Question not clear, or is the?
No, I'm just trying to. You add a reagent that inhibits Mek. That condition, the Mek levels are what?
Yes. Okay. That's the first thing to note. What is plotted is the abundance of phosphorylated Mek, but this reagent inhibits the activity of phosphorylated Mek. But So actually you have to, this is already sort of, yeah, so there are two things, the abundance and the activity. But at least, simplifying it again, U0126 does something with MEC. So then you would say that MEC causes ref because also the ref concentration changes. Right? Now, but the ground truth says that no, this is wrong. It should be the other way around. Ref should cause MEC. And this is in all the textbooks in biology. So it's probably the ground truth is correct here. Yeah, in the papers nowadays, you see the feedback loop. Ref, MEC, ERC, back to MEC. Back to ref. But so that's actually the question. How can we explain this disagreement? It could be that the inference is just wrong. You know, we just made a mistake, and we believe in the consensus. Or it could be the other way around. Maybe the consensus is just wrong. Inference was correct. Or there could be a feedback loop. Or also, that's what biologists often say to me when I talk about this. Maybe the inhibitor wasn't as specific as we assumed. So maybe U0126 also, in addition to changing something about MEC, also changed the ref. So, okay, it already gets a bit tricky, the validation here, if we want to do it more fine-grained, let's say. Now, this was one error. So here you see our reconstruction. And this was we got this arrow wrong, well, according to the consensus. But if you look in the data, so using Fisher's method, you would see that, if you do this, if you change MEC activity, then we see a significant change in ref concentration. So according to Fisher, there should be effects ref. And also the other relations or many of the other ones that are wrong according to consensus are very strong signals in the data. So what do you believe, the consensus or the data? So at the end of the day, conclusion, there seems to be a meaningful and to a certain extent reproducible causal signal. So many different causal methods, discovery methods find more or less the same thing. But that's nice. That's, I guess, a success. On the other hand, it's not clear what deviations from the consensus mean. Is it wrong inference, wrong consensus? What's going on? And validation is only possible up to some degree, because the ground truth may be unreliable or incomplete. Now, other data sets have come along. For example, Bodenmiller, you may have heard about before from Sofia Triantafilou. Mass cytometry data. This is much more complicated, extensive experimental design. Now there are 27 inhibitors times 12 activators times 8 dosages for the inhibitors. And we now measure, or they measured like 30 proteins. Nice dosage response curves. This looked very promising data to answer those questions that were remaining after looking at the flow cytometry data. So what did they do? They tried evaluating local causal discovery by Cooper, one of the nicest causal discovery algorithms around because of its simplicity. View of the time, I'll skip how it works. You don't know it's recommended reading. Here's an example actually of how an LCD pattern in the data might look like. Here we have again, it's a similar setup as before, so two conditions, green and red, baseline and BMA Yono activator added. And if you look at the data by eye, you already see these independent. Oh, sorry, see those? Yeah, you do so. You do see those for this particular LCD triplet. So you could conclude that S6 is causing ERK. Now we can do this without having to rely on background knowledge on what this activator is doing. So whether it's specific or not doesn't matter so much. So what did they do? They found several LCD patterns in the data. They used the CAG database as an external ground truth. And they found that some of the LCD predictions are in agreement and others are in disagreement. So, well, that's kind of the conclusion. And is this now successful, yes or no? I don't know. Because we don't know how accurate is the CAG database here. But also, unfortunately, this data set, if you look more closely, these LCD results that you get, they are not very robust. So contrary to the flow cytometry data here, things change depending on what causal discovery method do you use, which parameters how do you tune your parameters, etc. So I didn't find anything reproducible in there myself. I was involved. I collaborated with them for a year or so, and so we worked on this data for a long time to understand what's going on, but in the end, I couldn't find something reproducible there. So I would say in general, mass cytometry is really promising for validation purposes, but probably this data set was a bit too early. Finding out how this technique could be used properly. So the data seems to be too low quality to be useful for validation purposes. Also here, it looks, in their paper, it looks as if these dose response curves, as if they have all these nice dose response curves here. But that's only for one of these 27 inhibitors that it looks. The other ones sometimes go like this. You would expect some sigmoid curve. So, yeah. So maybe in the future, if a better data set comes along, we can revisit this. So, conclusion, discussion of this list of, so now I pose a more specific question, what are the convincing success stories? And, well, IDA is, me, no, not convincing. This one is also not convincing. Trigger, we still have to look into that one. We are currently looking at that, whether the validation that they actually performed is somehow convincing. We're trying to match it up with the Kemmeren data, because now we have two data sets that we can sort of match up, rather than having to rely on some bioinformatics database. Karen, so the flow cytometry data, is it convincing, yes or no? It probably depends on whom you ask. I would say yes, but you're not, yeah, you could also be skeptical and say no. ICP, for now, I would say yes, that's a success story. But it's also, of course, not an absolute proof. So are there really convincing success stories? Maybe ICP, but I hope we really will have those one day. I think that would be very good for the field. Try and get there. Thank you for your attention. And I would be happy to receive by email other suggestions for maybe look at this paper, because I really would be happy if we can find something really convincing. Who did that? You? Or? Oh, Steckhover, yes. I see. Yes.
And they claim
I forgot if we did bootstrapping. I think we bootstrapped ICP at some point. Bootstrapped. But that would be definitely worth, that would already help. For example, yeah, bootstrapping seems to help a lot in general. So worth trying, yes.
how much of the problem not having it round.
A big part of it.
The algorithm. So I mean, the ideas about.
So I think a big part. So this is really this chicken and egg problem. So in the past, when I had the postdoc, I had this. Well, for example, this data, the results I showed for the Sachs data set. I went to some biologists and said, look, here's this nice method. You can do experiments. Can we sort of collaborate, and we can write a nice paper together to find new causal relationships that are not unknown to biologists. The typical reaction was, oh, interesting, I've never heard of those methods. Maybe we can do that, but first I would like to see that it actually works. And then, for example, for the Sachs data set, but how do you know these inhibitors are so specific? So it was before I could even get to the point of convincing them to do measurements together. There was this. So in a way, there's the data collection is really expensive and it's a huge effort. There's yeast data, the Kemmeren data. They was a multi-year effort with a robot in the lab doing all these experiments. So maybe you have some better resources than I have to set up experiments. That would be great, because I think that's an important part of what's still missing. Because I think the algorithms, we might be actually quite close, but we just don't know, are the algorithms wrong and the predictions we get, or is it the ground truth that's wrong? With the interventional data, like the yeast data, the really nice thing there is that you don't have to rely on an external ground truth. It comes from some data process. Just look at the data itself, and it's all hopefully from one experiment under controlled conditions. That should be easier, I guess.
They do differently.
Well, in the Kemmeren data, actually they did it 4 times, and what is presented to us as one measurement was actually already they had 2 biological replicates and then 2 technical replicates, and they did some averaging and pre-processing, make sure outliers were removed and everything. So this is already an average, let's say, of 4 measurements. Also in also unfortunately in the Bodenmiller data, there's no repetition, for example. So whenever I talk to biologists, I always say about this team, I always say, just put in more redundancy for us, please. Put in more repetitions. For yeast, I don't know how quickly yeast grows. It's multiple generations of yeast. So I think it's days, I think.
From a single cell to
I think,
5% of data. But,
yeah, something like that.
Is that you might have a lot class, so this will give you positive observation.
Well, at least you would get to see the indirect causal effects on that time scale, let's say, for that system, right? So if you can match it with gene regulatory network that is somehow found in other ways, that's another question. But yeah.
And that's why I was coming. I will talk that. Second, so this is tentative values. You might have A affecting, and then there will be a D, and this one, the other one is activated.
Yeah.
This one is still.
Yeah.
Now, regarding to that, one of the earlier studies of Bayesian network.
So are you referring to the cell cycle data, Spellman cycle data? Yes.
Then in the external validation, data macro-arrays.
Yeah, so I wasn't entirely, maybe I should add it to the list, actually.
Right.
Just that I'm familiar with that work, but I'm totally, the validation they used says me nothing, so how convincing is, I don't know, but right. Probably it's worth looking at that also as well.
Where they look on.
Yeah, okay, so we also used that transcription, that Mek-Isaac data, for example. We also, actually, what we also did that I didn't tell you about today is we used these external, well, doesn't really matter. We also looked at some external ground truth. For example, transcription factor interactions.
Right.
And what we found is that the overlap with what we find here on this data, Kemmeren type data, is really very, very, very small.
That might be because they
could be, but it also suggests that maybe we should sort of, before I was naive when I started this, I thought, okay, we are looking for the gene regulatory network of yeast, but maybe there are many different ways of how you can define it, and maybe they all look very different.
Great talk.
Yeah.
Maybe I also have a question. And we'll do this discussion. most time the curve in G rather than the causality. So, and actually, if you have the system, it would be better if you try to learn the causality network by monitor not only the causal relationship. You notice it?
So I'm not sure I entirely understood the question.
Are you talking about learning from manipulation?
Yeah.
So
In a way, the ICP method is doing that, but it's doing it in a very. It could do it. It could use much. It could much better exploit the available information. Now it just says, okay, these are all kinds of different perturbations on the system, and we are looking for anything that remains invariant under all those perturbations. But if you actually know what perturbation you have done, which gene you have knocked out, then you can, of course, infer much more from that. And that is basically what I was referring to here. Shouldn't we be able to exploit that information and do causal modeling of that data in a much better way? And I was thinking about using constraint-based causal discovery, but then looking at these particular characteristics of this data. Okay, I can say I already tried. I tried LCD, and it didn't work better than random guessing. And I don't understand exactly why, because, well, okay, one reason why could be that there are so many faithfulness violations in this data for whatever reason that if we think we see a conditional independence, it still doesn't tell. Oh, is it faithfulness or is it? Well, we definitely have to assume faithfulness for LCD, so could be one explanation of why it doesn't work, but could also be measurement. There could be many things that are going wrong, but one problem is also the scale of this data. So with LCD, we looked at 3 variables at the time. Of course, if you take them all into account, you have a much better chance of disentangling the whole causal structure to make it look sparse. But I don't know any methods that could be applicable on that scale. Well, okay, PC or FCI could be, but then you have to first extend them to use the, well, no, okay. Anyway, it's a great proposal. I think people should look into it. I'm a little bit skeptical how well it's going to work based on my early attempts due to these numbers here.
Probably a very painstaking process to go through all these things.
Yeah, it takes a lot of work indeed. That's a very good question, and I have several. I don't know the answer to that. For example, for this data set, it seems that cycles are not so important here. If you have data where you expect there could be many feedback relationships, then maybe I could say maybe we have to reconsider how we actually model this in the first place. Because if you allow for cycles, then everything is much more complicated. No, this is population average data.
So
that's another problematic thing. In combination with the selection bias, these cells have to grow for several generations. Now, another reason why LCD might not work, that suppose we have something like this, and we see a conditional independence. Well, if we have selected on the survivability or the fitness of the yeast while doing this measurement, and it's somehow caused by both Y and Z, then LCD gets confused. And that this path, well, okay, we have to think about the true positives and the true negatives, etc. But I haven't thought this through entirely, but the selection bias here would really mess things up massively.
Because even if Y is not causing Z, X and Z are still independent of
Thank
Y.
you. That was the point. Yeah. So yeah, exactly. So So here we still see an LCD pattern, and we would conclude that Y is causing Z, but it isn't. Well, and then the question is, what is our definition of causality actually? Are we talking about population level causal effects that remain after growing the yeast for 7 days? And if we see a change happening there, do we call that a causal effect? Then still, the conclusion that Y causes Z might be valid, effectively speaking. But if you would say, no, that's not what we want to call a causal effect, because clearly it's just selected on this thing. And it's kind of, how accurately do you, well, basically it's like, do you treat this as a latent variable or not? I guess that's what it is. If you if you marginalize it out, then get but the funny thing is that this selection bias, you also see it in the type of experimentally, right? It's even if you do this randomized experiment here, then it's still there. So according to that, you would call it a causal relation. So that's one of the major complications to me for this data set. I really don't know what the selection bias, how it messes up our reasoning.
Lighter selection bias up by an unshift.
Yeah.
But theoretically, deal with that.
With this problem.
Yeah, because it wouldn't fool, right? You wouldn't take it. You had a W. If you were using a collider to orient rather than wouldn't find the collider. Wouldn't fool you. It would just say, not give you an answer.
I'm not so sure in which sense it wouldn't fool you, because it seems to depend on how you define because we also have these interventions here, right? So it really gets into the discussion of what causal effects are we talking about in the first place? Are we willing to call this a causal effect of Y and Z, even if it's not a direct effect, but
what
just...
I'm saying there is got X independent of pulls you into and you're oriented and you're using the orientation. Using the orientation.
Yeah.
As Y, Z. So oriented it as a non-collider.
Yeah, because maybe you know that Z doesn't cause X and doesn't cause Y. So one way to get it is use the acyclicity, just assume it, and if you see that X actually causes Z and Y causes Z, then it can't be the opposite way. So get this.
So I'm saying if you were you're orienting via collider rather than
okay.
wouldn't do it.
So if we would just look for Y structures rather than LCD patterns, the selection bias may not be a problem.
Right.
For example. Yeah, okay, but that's still something you could try doing.
Yeah, I think that might.
Okay, if that would help already, that would be worth trying. But so I think it depends. So to come back to your question, I think we should think more carefully about selection bias in data sets like these, about cycles in data sets like those, the signaling networks. But in the end, reality will always be more complicated than we think. Even we should hopefully at some point, without making all the assumptions, at least outperform some random or some simple baselines, some data set, right? To me, it's not either or. I think we should both think about more careful causal modeling in these and see how far we get by simpler approximation. Complicated your models become, the harder all this inference